Okay, thank you for all joining us and welcome to the Sepsis Canada Summit Series webinar. My name is Christine Russell and I identify as she, her. I am part of the communications team at Sepsis Canada. Today and throughout this series, our research teams will be providing an overview as well as progress to date on their current network projects. We will take about 5 minutes for quick introductions, housekeeping items, and following these, we'll begin our presentation. Please feel free to ask questions throughout the webinar today via the chat box. The chat box will be moderated for questions, and at the end of the presentation, we will answer them during the final few minutes of the webinar. During the presentation, all microphones will be muted. If you wish to ask a question at the end of the presentation, please feel free to raise your hand and we will enable your microphone to be unmuted. Lastly, just a reminder, this meeting will be recorded. We will be publishing it on our website in the news and events section for those who are not able to attend the session today. I would like to acknowledge the Indigenous peoples of all the lands that we are on today. While we meet on a virtual platform, I would like us to take a moment to acknowledge the importance of the lands which we call home. We do this to reaffirm our commitment and responsibility in improving relationships between nations and improving our own understanding of local Indigenous peoples and their cultures. From coast to coast to coast, we acknowledge the ancestral and unceded territory of all the Inuit, Métis, and First Nations people that call this land home. Sepsis Canada is a nationally coordinated research network with over 200 members and representation from universities and academic institutes across Canada. Funded by the Canadian Institutes of Health Research, Sepsis Canada brings together experts from various disciplines working collaboratively on interrelated projects under an integrated and unified program. Our mission is reducing the burden of sepsis for all Canadians. Sepsis Canada has created 3 teams within our network. And through the Sepsis Canada Summit Series, you will have a chance to hear from all 3 teams, which will include updates on their current projects, progress to date, and their next steps. And with that, I would like to introduce you to the speakers in today's presentation. As the co-lead for Team 3, Project 10, Dr. Manoj Lalu is an associate scientist appointed to the Ottawa Hospital Research Institute Clinical Epidemiology and Regenerative Medicine programs. He is also an assistant professor at the Department of Anesthesiology and Pain Medicine and a staff anesthesiologist at the Ottawa Hospital. Prior to medical school, he completed a BSc honors and a PhD in pharmacology at the University of Alberta. Our other co-lead in this project is Dr. Asher Mendelson, who is an assistant professor in both the Department of Medicine and Immunology at the University of Manitoba. Originally from Montreal, he completed a degree in health sciences and music at Marianopolis College. He attended medical school at McGill University and completed residency training in internal medicine at the University of British Columbia, as well as a master's degree in experimental medicine. His research focuses on microvascular physiology and sepsis in critical care using both preclinical and translational approaches. And last but not least, our last co-lead on this project is Dr. Braedon McDonald. Dr. McDonald is an assistant professor in the Department of Critical Care Medicine and Department of Medicine in the Cummings School of Medicine at the University of Calgary. Originally from Crossfield, Alberta, Dr. McDonald trained in microbiology and immunology at McGill University in Montreal, followed by MD as well as PhD training in immunology at the University of Calgary. He completed an internal medicine residency at the University of British Columbia, followed by a fellowship in adult critical care medicine at the Cummings School of Medicine. Dr. McDonald completed a CHR and AIHS-funded postdoctoral fellowship in microbiome research at the University of Calgary's International Microbial Center. Dr. McDonald is a clinician scientist at the Snyder Institute for Chronic Disease and International Microbiome Center, where he leads a translational and basic science research program on microbiome in immune interactions in infection and critical illness. Dr. McDonald's clinical practice focuses on multi-systems intensive care, and he is an attending intensivist in the ICU at the Foothills Medical Center, Rocky View General Hospital, and South Health Campus in Calgary, Alberta. And with that, I will turn it over to Dr. Lalu to begin their portion of the presentation.
Great, thanks so much for that introduction, Christine. Can everyone see my slides just before I start getting right into this? Good, okay. And thank you to Sepsis Canada for the invitation to share some of our work to date. So I'll be starting off this presentation with just a brief overview of the rationale for some of the work that we've been doing. And then Braedon will take over and he'll be speaking to how our National Preclinical Sepsis Platform fits into the landscape of Sepsis Canada, and also be overviewing a little bit of sepsis pathophysiology. And then finally, Asher will be capping off this presentation with a brief summary of some of our work to date and highlighting where we're going to be going next as well too. So in terms of the rationale and where this all arises from, part of this can be traced back to what's been termed the reproducibility crisis in basic science or preclinical research, or laboratory work, in other words. In terms of what's been happening over the last decade plus, people have been pointing to the fact that findings in one lab are very difficult often to replicate in another lab or a different center altogether. Or sometimes even the same findings from the same lab are often difficult to replicate. And it's even come to a point where top tier clinical medical journals are actually pointing to this issue and saying, hey, preclinical research needs to evolve in one form or another. And this is a very complicated issue as to what is actually underlying this reproducibility crisis. However, I will mention here just some very key issues, I think, that may be contributing to this. And these are some of the methodological challenges that we face when conducting preclinical laboratory-based research versus clinical research. So for instance, preclinical research is often single laboratory, single center, such as my lab, working often with ourselves and not really reaching out to Asher or Braedon's lab, for instance, versus clinical research, multi-center clinical trials, as most of you are probably familiar with, are very, very common. In fact, it's considered a gold standard. Methods to reduce bias like randomization and blinding are very rarely instituted in laboratory research. However, in well-conducted clinical research, where it can be instituted or applied, there is randomization and blinding and other methods to reduce bias. Third, there's very rarely sample size calculations in preclinical laboratory research versus clinical research. Often well-conducted clinical trials are adequately powered and they've pre-specified their outcomes as well. Finally, there's these variations in protocol that occur very frequently in the laboratory, making it even more difficult to reproduce. However, in clinical research, often you're sharing protocols. So that leads to, perhaps one solution that we can look at to address this reproducibility crisis, which is preclinical multi-center studies. So what are those? That's where multiple independent laboratories perform the same experiment using the shared protocol. And again, as I've already mentioned, this is considered a gold standard in many forms of clinical research. So in other words, here's my lab with our protocol. Well, why not share that with Asher's lab and Braedon's lab and Patricia's lab and Allison's labs, for instance, and see if we come to the same results. What's the level of variation between our laboratories, and ultimately how reproducible are these results? And that gets into the why of these multi-center preclinical studies. Again, we want to test the reproducibility and generalizability of our findings, and this is a great way to potentially do that. It could be a more robust way to approach our science than our typical single laboratory studies that we've been doing. And finally, one thing that's really great and that we've already experienced in the NPSP, or National Preclinical Sepsis Platform, is that you bring diverse expertise to the table. So it's not just my brain sort of leading my lab, but it's also all of a sudden I've got Asher there, and I got Braden, I got Alison and Patricia, and others from across the country that I'll mention in a later slide. So we're all coming together with our diverse expertise to try to solve an issue. So finally, because of all these potential advantages, we're wondering if this might be a way to increase the efficiency of preclinical translation. Could we be able to identify potential targets or potential therapies with greater accuracy before we start moving into early phase clinical trials? In other words, would we be better picking the winners? So in terms of what we've done, and I'll provide a little bit of an overview of our work to date, one of the first things we did, and this is a couple of years ago now, is actually look in the literature and say, hey, who else has taken this approach? Who else has taken this multi-laboratory approach? And, much to our surprise, it's actually been very, very few. And only 16 multi-laboratory studies have ever been conducted and published. And that's probably very surprising to those of you that conduct clinical research, because as you are well aware, there are hundreds, if not thousands, of multi-center clinical trials that have been conducted to date. But what's really interesting, though, is that 12 of these studies have been published more recently, so 2015 onward. So this does seem to be gaining some momentum, and many different clinical spheres being investigated, like stroke, brain injury, or myocardial infarction. However, what's interesting, again, is that no one's actually looked at sepsis in this sort of model, so making it even more exciting for sepsis to step into this. So just give me 1 second. I'm going to close my door here because I think there might be some background noise. Sorry, and I'm back. So in terms of comparing these single and multi-laboratory studies that we actually found, there were 16 multi-center studies, again, that we had found in the literature published, and we actually paired these to 100 single laboratory studies, and we compared different characteristics. I'm just showing you some highlights here. So first of all, really interesting, the median sample size, very, very different. So 111 in our multi-laboratory studies versus 19, which is very typical for a single laboratory study. The total number of animals being used, though, it was quite similar between these two types of studies. And in terms of the animal models that they're looking at, or at least the species that they're using, not surprisingly, they're using these rodent models, rat and mice models that were very, very common in laboratories. And one of the really interesting piece that we found here was that when we compared the rigor of these multi-center or multi-laboratory studies compared to these single laboratory studies, these multi-laboratory studies address these measures to reduce bias, like I've already mentioned, like randomization and blinding, sample size calculation. So they instituted all these measures to reduce bias and increase the rigor of their study far more frequently than these single laboratory studies. And perhaps because of that, we're actually seeing differences in the overall results that came out of these multi-laboratory studies versus their matched by species, and also the intervention they're looking at matched single laboratory studies. So what you're looking at here is a graph where the blue dots represent our single laboratory studies, the red dots represent our multi-laboratory studies. And the closer these dots are to this line that goes all the way down here, the smaller the effect size. So you can see over and over here, again, the trend that these multi-laboratory studies are much closer to this line, meaning that their effect sizes are much smaller than their counterpart single laboratory studies. And what's so interesting here is this is the exact same effect that we've seen in clinical studies. So as you increase rigor of your clinical studies, and as you move from single center clinical trials to multi-center clinical trials, you also see this exact same decrease in effect size, which may be discouraging, but also more often reflects what happens in real life as well too, when we actually start to apply these therapies to patients at their bedsides. We also went on to do an interview study, so a separate study where we actually reached out to these 16 groups that had conducted these studies to learn about how they did their studies and learn from them before we started our own multi-center preclinical study. And the barriers we found, first of all, over and over again, people were saying funding was an issue. Funders didn't recognize the value of this approach, but we've been very lucky, obviously, with Sepsis Canada investing in this. And there's also been an increase internationally in funding of these studies over just the last couple of years. The culture and climate of the preclinical science community, there's a lot of skepticism and misunderstanding about what a multi-laboratory approach actually is. Another difficulty was harmonizing the protocols between all these different laboratories. And then finally, differences in lab resources and ethics between these different centers as well too. In terms of facilitators, these different groups said, hey, what really worked well was open-mindedness and trust between these investigators that were involved in this multi-laboratory study, having regular meetings and engagement of all the different stakeholders. And then finally, site visits and actual training for the people at the bench who are doing and conducting this work. So that leads us to the National Preclinical Sepsis Platform. So taking all this information that we gathered from our interview studies and that systematic review that I just told you about, we came together with like-minded folks from across the country, and we came up with a set of objectives that we want to accomplish over the next few years. So the first is actually to identify and engage relevant stakeholders. Secondly, as Asher will be showing you, we're starting to develop a fecal-induced peritonitis model of sepsis, so a mouse model that all laboratories could potentially use downstream. And then we decided as a group that our first target of something we want to investigate was the differences between male and female mice in this case that have sepsis induced and that we've given antibiotics and fluids to. So that will be our first sort of target. And then finally, more downstream, we're hoping that we can use this model and everything we've learned about it and also this multi-center approach, obviously, to assess really new therapies for sepsis as well too. So in terms of what we've been doing, obviously we've had a very large group of stakeholders come together. I've already mentioned a few of the scientists that are involved in their laboratories, but also mentioned we've had veterinarians. We've had patient partners that I'll briefly mention in my next slide as well too. And other trainees and highly qualified personnel all come together, and we've formed a smaller working group, but we're still continuing to reach out to these other folks. And I think Braedon's going to be mentioning some of those as well too. We have 6 participating centers. Again, this is just for our core group. We're going to be using this fecal-induced peritonitis model that you hear a bit more about in later slides. And again, we have highly qualified personnel, HQP, veterinarians, and we've really tried to take this integrated knowledge translation approach where we have multiple stakeholders, not just Asher, Braedon, and I, and that's it, but really trying to get everyone across the country and different stakeholders involved in this. And we've been meeting monthly now for over a year. In terms of progress to date, I'll drop in the chat our first paper that we published, which is really a proceedings of a meeting that was funded by CIHR to bring all these different folks together. This is a picture of that meeting. It was in Niagara-on-the-Lake the summer before the pandemic. So one of my last meetings at that time before the pandemic. And but I encourage you to take a look at that. It gives a bit of an overview, again, of the rationale for doing what we're doing, but also tells you about where we want to head. Some of the studies that I've already showed you, that systematic review and the interview study, those papers should be forthcoming this year. And we've also been funded to actually do systematic reviews on that first topic, right? So I said, hey, we want to look at biological sex and antibiotics in these animal models. So we've done a systematic review in this area, actually a couple of different systematic reviews, and I'll drop those in the chat as well too if you're interested. But again, sort of taking the same approach that many of you are probably familiar with from the critical care trials group, so the clinical group, where they do a lot of this background work before they get into their first, large clinical trials, et cetera. So we're trying to take that same sort of evidence-informed approach as we go towards our first real multi-laboratory study. We've been funded generously, again, from Sepsis Canada. We've also received funding from the New Frontiers and Research Fund, federal funding. Our systematic reviews that I've mentioned have been funded by CIHR, and the Canadian Anesthesiology Society has also funded some of this work. And this is very much a platform, and we're looking for other ways to expand this platform as well too. So not only are we trying to conduct this first in Canada multi-laboratory study, but we're also studying the collaboration. How do we actually collaborate with each other? So this is a project that was funded by the New Frontiers in Research Fund, and Dr. Jenna Evans, who's at the business school at McMaster, is leading that. So she's actually interviewing all the different stakeholders who are involved and seeing how we collaborate and how do basic scientists actually work together. I've also mentioned our patient partner panel. So patient engagement, as many of you already heard of in previous seminars, is gaining a lot of momentum in clinical research, but it is very new, very, very new at what we do at the bench. So in other words, patient engagement in basic science is very much a cutting edge area that we're going to hopefully lead the area or lead as well too. We've been funded, at least my group's been funded by the Stem Cell Network to look at how we can actually engage patients in basic science. And again, the NPSP is going to act as a bit of a platform for some of this as well too. We've had our first few meetings with some of our patient partners. I see some of them online here as well too. And I hope next year when we give you an update, we'll have one of our patient partners actually present some of the work that they've been doing within our group as well too. Finally, we have a journal club coming up. We're trying to, again, make this very innovative. It's going to be trainee led by a couple of NPSP trainees, but we're also inviting the corresponding author to come talk to us as well too and be part of that journal club. And it's actually on a preclinical model of sepsis. So please join if you're interested. So this is a brief update. I'm going to hand it over to Braedon right now. I'll mute myself and he can take over.
All right, thanks, Manoj. So I'll take a few minutes here and talk about how this platform exists, integrates, and functions collaboratively within the Sepsis Canada network. And I'm not going to go into too much detail about the Sepsis Canada network because others on the line, including our Grand Poobah, Dr. Fox Robichaud, would know much better and in much greater detail how all of the platforms link together. But just to introduce and go over the NPSP's vision of how we can integrate within the Canadian sepsis research landscape. And so the Sepsis Canada network, of course, was funded by the CIHR under the recognition that sepsis research really needs a collaborative and integrated approach that incorporates research from the bench to the bedside and beyond, including within the public relations realm. And so the NPSP has really embodied this philosophy. And as Manoj has demonstrated, the work that's being done extends beyond just preclinical models of sepsis. And as a group, we're really trying to reach our tentacles into all of the different objectives of the Sepsis Canada network from the perspective of preclinical research. And of course, there are other platforms within the network that are doing the same within the clinical trials realm, advocacy, health literacy, knowledge transfer, and population health, and others. Next slide, please, Manoj. So the NPSP is under the umbrella of Team 3 within the Sepsis Canada network, which is focused on translational biology, as well as the concepts of clinical trials, because of course, one of the major objectives of the NPSP is to develop a preclinical sepsis trial platform. And the lead under this umbrella is Dr. Paul Koobs, here, a colleague here in Calgary, and our key knowledge user who also participates in the NPSP platform is Dr. Patricia Liao at McMaster, who's the lead for the Canadian Critical Care Translational Biology Group. And so, as Manoj has already gone through, the NPSP is being led not just by the three of us, but really by a group of collaborative scientists from across the country. And so as part of the Sepsis Canada network, part of the goal for the NPSP is to enhance the collaborativity of sepsis research in Canada and bring labs together that would in the past have been working in isolation. And so in terms of a national strategy to tackle sepsis from a discovery science and translational science perspective, it really fits well within the umbrella of the Sepsis Canada network. But of course, the other thing is that we're trying to develop a platform that can be utilized by the sepsis community. And in doing so, generating not only model systems and a network of researchers that sepsis investigators can eventually tap into to help augment, accelerate, and enhance their research in sepsis, but also by, for example, constructing a biobank of experimental samples from mouse research that can improve the efficiency of sepsis research and reduce the use of experimental animals and things like that. So lots of work going on to try and tackle some of the limitations, as Manoj outlined, in the current sepsis research landscape. And eventually, the plan is for much of the work that's being done within the NPSP to link into the human sepsis research realm, where, for example, novel therapeutics being tested and vetted within the preclinical realm can interact with the clinical trials platform within Sepsis Canada as a strategy to accelerate the translation of novel discoveries towards meaningful clinical benefit for patients with sepsis. And the Sepsis Canada network is set up to facilitate that accelerated translation through, for example, the interactions between our group and the leads within the clinical trial and clinical research realm, such as Dr. Francois Lamontagne and the knowledge user, Dr. Rob Fowler, who's the head of the Canadian Critical Care Trials Group. Next slide, please. So Manoj has already really encapsulated the rationale for why we think the NPSP will be of benefit for the sepsis research community and clinical care community. And just to reiterate, there is an unmet need to improve sepsis care. And this is something that's been covered in past presentations, so we won't go into it in too much detail. But the care of sepsis is currently a supportive care strategy because we don't have targeted therapeutics to tackle this entity as a disease. And in many ways, it's a one-size-fits-all approach to both the diagnosis and management. So there's a lot of work to be done to improve sepsis care, including right from the foundations of discovery science. And coupled with that, as was mentioned, there is a struggle to translate discoveries from the preclinical realm into the clinical realm within the field of sepsis and other fields. This isn't just a problem in sepsis, but it's prominent in sepsis because there have been so many therapeutics that have failed to move from positive signals in animal studies to meaningful benefit to patients suffering from this condition. And at the risk of not being too repetitive, I'll just say that this is partly because of methodological limitations within the preclinical realm, and those are things that we're trying to address, as well as the protracted timeline for translating discoveries from the bench to the bedside. It can take years and in some cases decades, and we think that's too long. And so the NPSP is looking to develop a way to efficiently and robustly vet novel therapeutics to bring them forward to clinical trials. Next slide, please. So before we get into the results of some of our pilot studies that Dr. Mendelson will present, I'll just introduce some of the concepts that are driving the work that's being done, and the disconnect between what we know about human sepsis and the animal model systems that are being used to study sepsis in the preclinical realm. So this table has a lot of information on it, but what you can appreciate just by gleaning what's in the right-hand column from the left-hand column is that the disease of sepsis in humans is a very heterogeneous disease, whereas the disease of sepsis as it's studied in animal models lacks much of that heterogeneity, because we're typically using genetically identical strains of mice. Most of the work that's been done has been done in male mice for a variety of practical reasons, historical reasons. Most of the animals that are studied are young, have no comorbidities, and there are limitations to the model systems, which I'll touch on in a little bit more detail in the next slide. In addition, the disease that unfolds in animal models has historically been a very hyperacute disease without investigating the more long-term impacts of sepsis that we now appreciate are very disabling for patients who survive the acute phase of the illness. And this is in stark contrast to the disease that unfolds in humans who are genetically diverse. It affects males and females, young and old. There are comorbidities that have meaningful impacts on how sepsis unfolds. And the disease can be triggered by infections across a variety of organ systems, as well as a variety of different pathogens. And humans, at least humans that live in areas of the world that have access to care, receive treatments of different kinds. And mouse models typically ignore the fact that these treatments are incorporated into the care of sepsis patients. So there is a limitation in what we would say call the construct validity of most preclinical animal models, and this is something that we're trying to tackle as well. Next slide, please. And how are we trying to tackle this? Well, in developing our models, we're trying to be holistic in the considerations of these factors that have limited the construct validity of animal models in the past. And it's by stepping back and really appreciating the fact that the pathophysiology of sepsis has many moving parts. But the key components, as we've identified them, are the pathogen causing infection, which triggers sepsis, the immune response of the host who's suffering from sepsis, as well as other host factors that can modulate how the pathogen behaves and how the immune response unfolds. Next slide, please. So in terms of sepsis pathophysiology, the studies that we're working on take into consideration the fact that there can be diversity of pathogens causing infection, and we are working with model systems. So that's important to consider, is that our model systems are just modeling sepsis. They're not meant to be comprehensive across the entire spectrum of what can possibly trigger sepsis. But it is important to consider the fact that when we talk about problems with reproducibility of previous research, if you really drill down to the details, often the pathogen insult is very heterogeneous, and in some cases even rather, I don't want to say inappropriate, but limited in scope, in terms of not even triggering sepsis with a pathogen, just a component of a pathogen that doesn't even replicate in the host. Immunological considerations are something that we intend to study and will certainly incorporate within the the rationale for how we develop our models, acknowledging the fact that the immune response unfolds in different ways at different stages of the disease, where the early phases of sepsis are characterized by a hyperinflammatory response, where the immune system is desperately trying to tackle this overwhelming infection, whereas the subsequent and later stages of the disease, the immune response becomes dysregulated in a manner that can actually put the host at risk for protracted organ dysfunction and subsequent infections, which in hospitalized patients would be hospital-acquired infections. And then considering components of the host response that we are now coming to appreciate can critically modulate not just the pathophysiology of disease, but also how therapies may or may not be effective, including the host microbiome and aspects of cellular and organ system function. Next slide, please. So this diagram is not meant to be something that we go through in detail, nor is it meant to be something that intimidates, but just to give a visual of the fact that there's many moving parts, and that as preclinical sepsis researchers, we have to concern ourselves with the complexity of disease and avoid the tendencies of the past to be very, very reductionist, because in doing so, this is what contributes to limitations in reproducibility of findings and translating findings to the much more complex situation of human sepsis. Next slide, please. So we've embodied and utilized this PIRO model concept for modeling disease, which was developed and conceptualized by Dr. Marshall. And I won't go through it in detail, but just to say that our experimental models that are going to be the foundation for this platform will take into consideration predisposing factors, as well as the insults, host response, and impacts on organ function and outcomes that all contribute to the modeling of sepsis pathogenesis. And in doing so, we hope that our preclinical research will be more effectively translated and more applicable to improving sepsis care in the clinical realm. Next slide, please. And so the general overview, although this is far more simple than what's actually being done, is that in our first cycle of experiments, as was mentioned by Manoj, the key aspect of the host response that is going to be incorporated into our study questions is biological sex, because we're learning from other realms of medicine, including cardiovascular disease, where it's now well appreciated that research that was historically conducted primarily in men to discover treatments that were very effective, again, primarily in men, are perhaps not effective or suboptimally effective in women. And we don't want to have that realization 10 or 15 years down the road. Instead, we'd like to incorporate those concepts right from the beginning and make sure that we understand what the impacts of biological sex are on disease pathophysiology, as well as the effectiveness of novel therapeutics. And one of the ones that we're going to focus our initial round of study on, based on a shared interest amongst a number of people in the platform, is intravenous DNase therapy, targeting a mechanism of pathogenesis involving neutrophils. Subsequently, obesity and or aging will be incorporated, and we'll take this as a stepwise approach. Next slide, please. So again, the systems and platform that we're developing are intended to be just that, a platform that the sepsis community can utilize. And many of us have already begun integrating NPSP experiments within our grants. The NPSP has provided and is happy to continue to provide letters of endorsement for grant applications. And it's been looked on very favorably. In my own experience, we did a CIHR team grant in chronic inflammation, looking at the longer term impacts of sepsis. And the reviewers really looked favorably on the fact that we were planning to use the NPSP platform to take novel discoveries and put them in the hands of multiple laboratories to accelerate the translation and hopefully improve the reproducibility of the experiments. Next slide, please. So now I'll pass it over to Dr. Mendelson to go through some of the results of our pilot experiments.
Thanks, Braedon. Thanks, Manoj, and thanks everybody for the opportunity to share some of our pilot updates today. So the NPSP, the first model that we're using is a fecal-induced peritonitis model in mice. This model was selected given its ease of reproducibility and standardization. This is compared with a number of other abdominal sepsis models in the preclinical setting. Building on the need for reproducibility, we felt that this was a relatively operator-independent setup. There are a number of time points looking at the evolution of disease progression that we're trying to address, starting with the hyperacute phase, which is a very short early inflammatory phase, and then moving towards acute and subacute time points. And as I'll show, we actually started with the subacute time point first and are working our way backwards. And that was in part to evaluate the feasibility of these models and incorporating clinically relevant treatments such as antibiotics and fluids. Next slide. So the objectives of this NPSP model were to develop a fecal-induced peritonitis model that exhibits a 20 to 25% mortality at 72 hours. And this was consistent with estimates from human sepsis. We evaluated a series of outcomes, including the Murine sepsis score, which is a clinical assessment of disease severity, as well as organ dysfunction and pathogen host interactions as discussed by Dr. McDonald. And then the purpose was also to determine if there were any observable differences between males and females in disease pathophysiology, as well as to design a biobanking strategy to harvest important organs for future analysis. Next slide. So this pilot study was developed in an iterative fashion here. This slide is not intended to be read by the people on the seminar, but it was broadly speaking, just to outline that what we did was pilot tested this design of the 72-hour FIP model at 2 sites out of the 6 of our core group, and that was McMaster and Ottawa. And as we evolved throughout each of these pilot studies, we manipulated various components of the treatment in an iterative fashion, whether that was the dose of the fecal slurry that was administered, or whether that was the timing of the antibiotics as well. Next slide. So the experimental timeline involved inoculation of the mice with fecal slurry at time 0, and then followed by supportive care with antibiotics, fluids, and analgesia at various time points, mostly every 12 hours throughout the course of the 72-hour experiment. This was done in consultation with the animal ethics committees across all 6 participating sites to achieve some consensus and standardization, as well as in consultation with our veterinary partner, Janet Tsunohiro Nielsen, who is working at Western University. Next slide. One of the things we discovered early in these pilot testing is that there appears to be a dichotomous result in terms of outcomes for mice in these studies. So these are mortality curves or survival curves. And really what we found, number one, is that there is a dose-dependent disease severity that can be observed with escalating doses of fecal slurry. But instead of being able to achieve this target kind of Goldilocks mortality in the 25% range, what we were observing is either there was very severe mortality with higher doses of slurry, or almost non-existent mortality with lower doses of slurry. And this appeared to be a feature of the model that was consistent through multiple iterative tests, as you can see here. Next slide. One of the other things that we manipulated was the administration of early versus late antibiotics. And by early, we meant within 4 hours of inoculation, and versus delayed antibiotics at 12 hours. And what we show here is that when you administer early antibiotics in this model, you essentially rescue the animals completely from achieving sepsis. And this was observed not only in their survival curves, as you can see here, but also in data that we're not showing, which was in bacterial translocation and bacteremia in the animals. So early antibiotics, not very helpful with achieving this construct validity that we talked about in human sepsis. And this is probably true because, when your appendix bursts, for example, you don't get antibiotics immediately. Typically, you're unwell at home for a period of time before you present to hospital. And this is maybe one of the reasons why preclinical models weren't necessarily translatable, is that oftentimes they were receiving antibiotics fairly early in the model. And so this is something that we show here needs to be addressed. Next slide. We then proceeded to, after gathering an enormous amount of data at McMaster University, repeating these studies at the University of Ottawa, and this was done to assess reproducibility. And interestingly, through a stroke of luck, we also were assessing a degree of how the slurry itself affected disease severity. So we had planned to use the same batch of slurry between both sites, which was always the plan for standardization, but through, as I mentioned, a stroke of luck, we actually changed out one of the pilots with a fecal slurry batch that was produced in an earlier year. And this allowed us to kind of evaluate how that moved forward. So next slide, please. So we noticed two things. When we used a slurry from a different batch, instead of getting a similar mortality to what we got in Ottawa, which at McMaster, which was kind of 85% versus 60%, so similar disease severity, we found that when we used a different batch, we got absolutely 0% mortality or 100% survival. And this was an unintended observation, which led us down a series of evaluations. And this was done in coordination with Dr. McDonald's lab at Calgary, who does some microbiome research. So we wanted to understand what was the composition of these different batches of slurry. Next slide. And so very quickly, what we're seeing here is we ran a sequence, not of the two batches that were used in this experiment, but actually two different batches, one from this experiment and one from a batch that was done at a different center. And we did essentially a microbiome sequencing of these different slurries. And what we show here is that although, generally the compositions are similar, there are clear differences in terms of how they're quantitatively distributed between the slurries. Next slide. And when you look at microbial diversity, as well as a principal component analysis evaluation of these microbiomes, what we're able to show is that there are clear distinctions in terms of the microbial composition between these animals, between these slurries, And this is demonstrated by differences in microbial diversity and the ability to segregate the slurries from McMaster and the University of Alberta. Next slide. So After having gone through those pilots, the last few slides here show that we're getting a similar dose dependent disease severity that was observed at McMaster. We're able to recapitulate that in Ottawa. And similar to what we observed at McMaster, there's really this dichotomous outcome of either very high mortality or very low mortality in these animals with no sweet spot in between. Next slide. And one of the things that we found that as opposed to trying to, observe these animals in a controlled environment and exit these animals when they reach clinically important endpoints, over the course of the study, there were a lot of unintended exits from the study in terms of animals that are being found dead instead of reaching their endpoints. And that's what's being shown here briefly. Next slide. And what we show here in terms of our scoring system, which was done as an intended method of evaluating these animals as they exit the study, we found that the murine sepsis score and even temperature to a certain degree did not predict which animals reached endpoint and which animals were found dead. So although the murine sepsis score is a good evaluation of disease severity for animals that are still alive, we found that this is probably not a good way of using it as an endpoint as a surrogate for mortality. Next slide. And we show here, for example, that there really wasn't a good way of predicting murine sepsis score or temperature to predict mortality. Temperatures of those animals that were found dead versus those animals which reached endpoint were very similar. And the temperature and the murine sepsis score of those that reached endpoint obviously was higher because that was built in as the endpoint, but the animals that were found dead prior to that had very acceptable murine sepsis score averages. So that wasn't a good predictor of them being found dead at future time points. Next slide. So overall, the challenges with this model were at 72 hours. This was a very onerous protocol in terms of around the clock, every 4-hour monitoring throughout the night and throughout the day for the labs. As I mentioned, the murine sepsis score does not appear to correlate or predict mortality, with many animals being found dead instead of reaching endpoint. The fecal slurry itself had a dichotomous outcome with either severe or absent mortality and a batch-to-batch variability. So in terms of how this fits with what we are intending to do with the NPSP model, we want to demonstrate feasibility and reproducibility and generate a clinically relevant model that can be used in all labs. So having evaluated the subacute model, we've gained insight into this, and we've now subsequently pivoted towards initial studies where we're going to be evaluating the hyperacute model. This is not intended to be the last model that we use in the NPSP, but is intended as a first or as a next step, which is going to be quite feasible and reproducible. Next slide. So this experimental timeline is much more truncated and is going to involve an inoculation at time 0 and followed by an evaluation of the animals at 4 and 8 hours. And this is going to be done in order to simulate not the evolution of sepsis or the treatment of sepsis in the intensive care unit, but really what the presentation of septic patients would be to the emergency department prior to any treatment whatsoever. And this is going to be done in order to understand what that initial hyperacute phase of sepsis is going to be and what either biomarkers or organ dysfunction we can observe in this hyperacute phase that may provide insights for how we evaluate, stratify, and diagnose sepsis in the emergency department for our patients in hospital. Next slide. And so very quickly, what we're showing here is that we are observing drops in temperature for these animals even early in the disease process. So we are able to mimic a septic and an inflammatory response at both 4 and 8 hours. Next slide. And we're also able to observe an appreciable increase in disease severity for these animals at both 4 and 8 hours, as evaluated by the Murine sepsis score. Next slide. So, in summary, the 72-hour model was difficult logistically, but we learned quite a bit about implementing multi-center studies and how we want to evaluate our preclinical models of sepsis. And importantly, we are able to show that our multi-center framework is feasible and reproducible between at least 2 labs and subsequently evaluated in multiple labs. Next slide. And the 8-hour model appears much more feasible with ongoing work that will further characterize in single and multi-center studies. So with that, next slide, we'd like to thank all of the participants. Oh, excuse me. Frequently asked questions for those that are interested. If I'm not part of the core group, am I still part of the NPSP? The answer is yes, of course. And we'd very much like you to continue working with us as we're moving these experiments forward. If not part of the core group, we have data that we are going to share with all members of Team 3 that they can incorporate into their own studies, as Dr. McDonald had alluded. And so what types of hypotheses are we testing? Mostly broad questions that can be interrogated with a multi-center format. And the hopes are that focused questions are going to be led by investigators within Sepsis 3 who can delve into very hardcore details in terms of mechanisms and pathophysiology, as well as rely on some of the biobank tissue that we're using. And if you want to get involved with future studies with us, please submit a one-page proposal of your program, and we can link you with investigators on the platform. Next slide. So we'd like to thank all of the members of the core group and a number of people. This has taken an enormous amount of work by a lot of dedicated professionals, investigators, highly qualified personnel, and others. And we're very excited to share this work with you, and we're excited to continue working within Sepsis Canada and the broader sepsis community. With that, we'd like to take any questions in the last few minutes here.
I think Alan had a question here. So just to briefly summarize, he was asking if we're accounting for the effects of clustering within our analysis approach. So Alan, I've talked about this with Monica Talliard, who's one of our methodologists here in Ottawa. She routinely runs cluster randomized trials. And it gets even more complicated because there's not only the effect of clustering at the lab level or the center, if that's what I call it. There's also effects of potential clustering because these animals are actually housed together as well too. So there could be cage clustering effects as well too. So for the first couple of studies, they're really going to be around feasibility more than any in-depth analysis in terms of what you're asking there. However, as we move towards a more well-designed large study where we're going to be looking at biological sex, then these are things that we're definitely going to have to consider in our analysis. And that's beyond the expertise of either Asher or myself or Braedon. So that's where we're going to get again other folks who are much more knowledgeable in this area to be involved as well.
Okay, thanks. I just wanted to check that this was being done or considered.
Yeah, for sure. And it's going to be really interesting because it's really new territory. I can say actually, one thing around that question though, like of the studies that have looked at this in the past, no one's actually really accounted for that in their analysis. There's a new group that was just NIH funded that just published their first study in stroke. I have to look through their statistical analysis plan. The study that they published was not all their results, but just more of their logistics and setup. And so it'll be interesting to see if they've actually thought around this issue.
Yes, very few human randomized studies have done this. And I'm not talking about cluster randomized study. I'm talking about individual subject randomized studies. There's still the issue of the possibility of similarity of outcomes within centers due to differences of care within centers or between centers. So this argument has been made, and there are examples of studies that have been reanalyzed, RCTs that have been reanalyzed using mixed effects modeling that have found different answers. So it is an important issue, and it's easy to do. It's easy to fail to do, but there's no reason to fail to do it because it's a relatively straightforward way to do it. It's a little more complicated in terms of the analysis, but it's no harder to do using a statistics package.
Sure. So it's definitely something we'll have to consider. And maybe I'll reach out to you offline for some of the examples of the reanalyses that you're talking about. So that might help point us in the right direction.
Okay.
Anybody have any other questions?
I just wanted to say Marie Maxime is one of our the patient partners for the NPSP. So thanks, Marie Maxime, for joining today. Like I was saying, maybe next year you can present. We'll have some interesting work that we can all present together.
I'm looking forward to that.
Okay, great. If there's no further questions, on behalf of Sepsis Canada, we want to thank you for joining us today. And of course, Dr. Lalu, Dr. Mendelson, and Dr. McDonald for their presentation on the NPSP. Our next webinar is September 15th. We'll take a break for the summer from 12 till 1 p.m. Eastern Standard Time. We'll be joined again by members from Team 1. I'll have more information released closer to the webinar date. I did put the link for that webinar in the chat box for anybody that wants to pre-register now. And again, like I said, there will be some more information on the actual topic coming out closer to that date. But thanks again, everybody, for joining, and have a great summer.
Thanks.